Hi, my name is Lydia Galvin, and for my final video project, I will be presenting on the article Nigrostriatal and Mesolimbic D2-3 receptor expression in Parkinson's disease patients with compulsive reward-driven behaviors. First, we must ask the question, what is Parkinson's disease? Parkinson's disease is a neurodegenerative disorder based in the pars compacta of the substantia nigra. Symptoms arise from the death of dopaminergic neurons over time in this region. Neuronal changes can begin up to 20 years before the onset of symptoms. It is most well known for affecting movement, but it can also have effects on sleep, mood, the GI tract, cognition, and speech. The most well-known symptom of Parkinson's disease is the resting tremor, one which disappears with action. A hand tremor can also be noticed while walking, and stiffness or freezing of gait are also common. The decrease in dopamine production affects the corticobasal gangliothalamic cortical loop, by muting activity in the direct pathway and enhancing the indirect pathway, resulting in inhibited thalamocortical drive. As a result, unnecessary movements are not dampened and will thus occur, most noticeably as tremors. We also have to ask, what are compulsive and impulsive disorders? Impulse control disorders are ones in which the patient exhibits an inability or failure to resist urges or impulses. A compulsive disorder is one consisting of compulsive actions or repetitive acts that feel necessary. A well-known compulsive disorder is OCD. Changes in signaling along the nigrostriatal and mesolimbic dopamine pathways have been shown to play a role in impulsive and compulsive behavior. It has been shown that patients who struggle with substance abuse have reduced D2-3 receptor binding, and that decreased D2-3 receptor expression in the ventral striatum has been implicated in the presence of impulsive behaviors. Increased impulsivity has also been found to correlate to decreased D2-3 receptor binding in the ventral midbrain. Dopamine agonists such as ropinirole and pramipexole are used to treat movement disorders such as Parkinson's. They have an affinity for D2-3 receptors and have been known to induce impulsive and compulsive behaviors. One study found that in Parkinson's patients with impulse control behaviors, there are reduced baseline levels of D2 and D3 receptors in the ventral striatum, due either to decreased receptor expression or increased ventral striatal extracellular dopamine levels. Another study noted that there was increased binding in the anterior cingulate and midbrain of patients with ICBs during a gambling-based task, but this only accounted for extrastriatal D2-3 receptor levels, not binding estimates. This study aims to provide an accurate estimate for binding levels in both striatal and extrastriatal regions, allowing them to quantify D2-3 receptor levels, or non-displaceable binding potential. It will assess these levels in many areas of the reward circuit of the brain, such as the ventral midbrain, striatum, and orbitofrontal, prefrontal, and anterior cingulate cortices. The researchers hypothesized that there would be reduced D2-3 non-displaceable binding potentials in the ventral striatum of Parkinson's patients with ICBs. The study consisted of 35 participants diagnosed with Parkinson's who were receiving dopamine agonist medications to manage their symptoms. Roughly half exhibited ICBs, a classification determined by an interview with the patient and the completion of the Questionnaire for Impulsive-Compulsive Disorders and Parkinson's Disease Rating Scale, also known as QIP-RS. The image on the screen shows how the participants break down into other categories and addresses which ICB symptoms each express. Falipride is a high-affinity D2-3 radioligand used to quantify D2-3 receptor levels. A structural MRI and falipride PET scan were performed during an off-dopamine state, one in which the patient's medications were not active. PET scans were performed on all regions of interest, or ROIs, including the caudate, putamen, globus pallidus, ventral striatum, amygdala, midbrain, thalamus, and cerebellum, with the cerebellum being used as a region of reference due to its relatively low D2-3 receptor expression. The study found that falipride binding was significantly lower in the bilateral ventral striatum and putamen of the ICB group. In A and C in the image, we can see coronal and axial brain slices from one patient, with A showing the ventral striatum and C showing the putamen. B and D show the mean non-displaceable binding potential for falipride in these regions, with significant differences noted in the mean across the two groups, which are ICB positive and ICB negative in both regions. The ventral striatum has been found to play a crucial role in reinforcement learning and addiction. The study's findings are consistent with previous studies that reported decreased baseline D2/3 non-displaceable binding potential in the ventral striatum of Parkinson's patients with ICBs. This shows that lower D2/3 receptors in this area have a clear association with the development of behavioral changes in patients with Parkinson's. Closer examination shows that in the ventral striatum, these receptors localize to the presynaptic mesolimbic terminal autoreceptors and postsynaptic indirect pathway medium spiny neurons. As a result, decreased D2/3 receptor levels have a clear association with decreased function of the indirect pathway, which mediates behavior change and avoidance learning. It is important to note that within the direct pathway there are no reported changes in D1 receptor expression, suggesting that, quote, when an imbalance between the direct and indirect pathways in the ventral striatum is coupled with increased dopamine release in response to rewarding stimuli, increased reward motivation in the setting of decreased avoidance learning could lead to the manifestation of ICBs and Parkinson's. While we are able to see a clear relationship between a decrease in D2/3 binding and behavioral changes, the question of whether this decreased binding precedes or follows dopamine treatment still remains. The images above show graphs comparing QIP-RS scores with midbrain valipride non-displaceable binding potential, with A representing the ICB positive group and B representing the ICB negative group. The results show a statistically significant positive correlation between midbrain binding and QIP-RS scores in patients with ICBs. This positive correlation was not noted in any other regions of interest. In healthy humans, impulsivity shows a positive correlation with anteroventral striatum dopamine release and a negative correlation with ventral midbrain D2/3 receptors. Parkinson's patients with drug-induced gambling addictions have shown increased ventral striatum dopamine release during gambling tasks. Increased dopamine release in this area has also been reported during visual cues relating to ICBs in patients with ICBs. This suggests that the neurological presentation of impulsivity in young, healthy humans and Parkinson's patients with ICBs share a common association with increased dopamine release in the ventral striatum. In healthy individuals, there is a negative correlation between midbrain D2/3 receptor binding and impulsivity, which makes a positive correlation between midbrain receptor binding and QIP-RS scores in Parkinson's patients unexpected. This can be explained by the fact that the number of D2/3 receptors in the ventral midbrain includes receptors on axons innervating the dorsal striatum, meaning that only part of the D2/3 receptor quantity is represented in this study. The researchers posit that if the number of receptors on each neuron remains constant between patients with and without ICBs, then the positive correlation may be due to more intact dopaminergic innervation of the ventral striatum. The major takeaway from this is that the positive correlation of D2/3 receptor levels with drug-induced behavioral changes suggests that Parkinson's patients with ICBs have a relatively greater preservation of ventral striatum dopaminergic innervation than those without ICBs. These images show valipride non-displaceable binding potential in the midbrain compared with other brain regions involved in the reward circuit, such as the caudate, putamen, amygdala, and globus pallidus, in Parkinson's patients both with and without ICBs. As shown on the graphs, there is a significant positive correlation between non-displaceable binding potential in the midbrain and in these mentioned areas in patients with ICBs. This positive correlation can signify many things, such as similar D2/3 receptors across these regions, coordinated influence of nigrostriatal and mesolimbic dopamine release or drug effects on D2/3 receptor expression, or the relative integrity of midbrain-based dopaminergic networks. This pattern occurs both in healthy humans and Parkinson's patients with ICBs. Previous studies have indicated that dopaminergic projections experience different rates of degeneration in Parkinson's, with loss of dorsal striatal innervation occurring before loss of ventral striatal innervation. This denervation, interestingly, does not always affect cells regulating dopamine release to the dorsal striatum, globus pallidus, or amygdala, something which could play a role in the development of ICBs. It thus appears that ICBs are associated with greater functional preservation of the midbrain and its dopaminergic projections. This greater integrity of midbrain dopamine and extended reward network, when combined with decreased D2/3 receptor levels in the ventral striatum and putamen, may cause a dopamine quote-unquote overdose from dopamine agonists in the reward system that drives the development of compulsive reward-driven behaviors and a lack of inhibition.